Welcome to this introductory video on how to analyze data within the software AltAnalyze. Today we're going to focus on both bulk and single-cell RNA-Seq data. AltAnalyze can process a number of distinct platforms, including microarrays, RNA-Seq, and proteomics data. There are a number of default workflows within AltAnalyze, and there are also vignettes which you can run individually. Today we're going to run a workflow that focuses on bulk gene expression analysis. Then we're going to navigate those results. And then we're going to run the default single-cell workflow. An important note is that no prior data analysis experience is required for any of these. Although AltAnalyze can work with relatively unprocessed data, such as the results of top hat analyses, including junction.bed files and exon bed files that are derived from those, here we're going to analyze a data set that's already been generated, in this case from a paper, single-cell RNA-Seq profiling of human pre-implantation embryos and embryonic stem cells. This is the supplemental data. We've already downloaded it. These are RPKM values for oocytes, blastocysts, embryonic stem cells that were derived from those blasts, including gene symbols. This is our primary identifier. We're going to save this as a tab-delimited text file on a folder somewhere, and then we're ready to analyze it. But first, we need to download AltAnalyze. To do that, we go to altanalyze.org. There's extensive documentation on this website, including manuals, tutorials, news and updates, a wiki, an FAQ. There are a ridiculous number of additional pieces of information here you can use. There are standard installers for different versions of the software. There are also development versions. Here I'm going to go to the development version. It's at SourceForge, and download it. Once you've started AltAnalyze, select Begin Analysis. You'll be prompted to download a species database. Select Continue. In this case, we have a human data set. There are multiple versions of the database available. Here I'm going to select human. For other databases, there's many other species available. There's up to 60 different species available. So I select human and continue. Once that database is downloaded, AltAnalyze will be ready to analyze data for a number of distinct platforms. If you want to perform RNA-Seq analyses, however, once you hit Continue after selecting RNA-Seq, you'll be prompted to download that database as well. I've already downloaded that database, so I won't be prompted here. And in fact, I'm not going to select RNA-Seq as my major data type, platform type here. Instead, I'm going to select Other, because this is an already normalized data set. RNA-Seq here applies to AltAnalyze raw process data. So instead, after selecting Other, I'm going to select a specific identifier that I know this dataset has. In this case, this is gene symbol. I've selected gene symbol, Homo sapiens, Other ID, and now I can select Continue. It's now going to prompt me to select Process Expression File or Additional Analyses. We'll come back to the second option later on. I'm going to select the folder that contains my expression file. In this case, I have this expression embryo.txt file. And the output directory is this single-cell embryo directory, which I've already created. Now, I could have also selected the text file that was in here, and it would have created some of these other subfolders. Next, I know that my data is not log. It is actually non-log data, so I'm going to select non-log. The rest of these default options are fine. There's a number of analyses supported within AltAnalyze that you'll see include cell type prediction analysis, expression clustering and visualization, QC, predicting biomarkers, and even batch effects removal if we wanted to. By default, it's going to perform a pathway enrichment analysis on a number of distinct resources. I can also set the stringency of these analyses. So instead of a raw p-value, I'm going to say that this has to be a Benjamini-Hochberg adjusted p of less than 0.05 to be significantly reported, and a twofold gene expression difference. For pathway analysis, I can select all the different pathway and gene set types to be analyzed here. I could also select an individual one if I wanted to. And I'm also can select to visualize overrepresented Wiki Pathways. This is a really cool feature that you'll see is really useful later on. Next, again, I've analyzed this data set before, so all of these data types are filled in here, but we basically assign a group name, and normally these are blank. And so we type in oocyte or paste here, for example, because we know these are oocyte samples, we know these are zygote samples, etc. And we can also establish comparisons here. So here I've already set late blastocyst versus 8-stage embryo, cell embryo, and zygote versus oocyte. And once we do that and hit continue, we're going to be running our analysis. So this will take somewhere on the order of 10 minutes to an hour, depending on what default options we've selected. This analysis is probably going to take somewhere on order of a half an hour. And so I'm going to skip ahead now. Once the analysis is complete, a number of graphical outputs will be reported to the user. These include quality control plots. These are showing the expression values binned for each and every sample, principal component analyses, hierarchical clustering results. These are the differentially expressed genes in our comparisons for each of the samples. Cell type prediction analysis, or lineage profiler. Here I clicked on human embryonic stem cells, and indeed these look most like human embryonic stem cells on average. And even what's called a marker finder analysis. And the marker finder analysis is trying to identify genes that are largely restricted to one group of cells versus another. In this case, we can see the oocyte restricted genes here on the far left. These are colored based on this legend here, versus the zygote specific genes, the 2-cell stage genes, the 4-cell stage genes, the 8-cell stage, and so on. And so you really can identify what genes are ideally the perfect markers for further validation. There are more results in this data plots folder in terms of graphics, and you also have a link to the results folder here. If we click this, results folder will appear, and there are a number of directories here that were created by AltAnalyze. For example, if we go to the expression output directory and find this file, dataset embryo, our dataset was called embryo, and open this in Excel, we will have access to all the fold changes, statistics, adjusted p-values, analysis of variance statistics, annotations, whether the genes are protein coding versus non-coding, what are the putative microRNAs that are found in the 3' UTRs of those genes, what algorithms are predicting those, where's the localization of the gene, is it in does the protein in the transmembrane, is it a transcription factor, is it a splicing factor? A lot of these are custom annotations that have been included on AltAnalyze. The raw expression values are also included here along with the averages. As I mentioned before, the log fold, fold change, unadjusted p-value. This is a moderated t-test, which is basically a lemma p-value, a lemma t-test p-value using an empirical Bayes moderated t-test. A Benjamini-Hochberg adjusted p-value. And at the far right, we can find our global group comparison statistics, such as our analysis of variance, p-value, and adjusted p-value, and the maximal fold change that was achieved. So this is a very useful output, and there's more files like this. There's a summary of all the changes in general, for example. You can usually open this in Excel, but I'll just open it as a text editor. You can see the numbers of genes changed for the different comparisons we had. And there's this folder called GoElite. Now this is a very useful folder for further comparison, for further analysis. For example, if we now look in the regulated folder, we can find all of the enriched pathways from our analysis if we open up this pruned results z-score file. So these are the different comparisons on the left, blastocyst versus 8-cell embryo. We have predicted cell types that are enriched. Surprisingly, this is 6-cell stage embryo from a previous compendium, and 8 to 10-cell stage embryo were highly enriched categories. So along with oocytes. So this is looking like very good data. Our algorithms are predicting things correctly. We have a number of other categories that we can look at: disease ontology, gene ontology, transcription factor target predictions, and much, much more. These are also available as graphs. So if you look under networks in this folder, you can actually view gene graphs that show which genes are associated with each category. So again, the 8 to 10 stage embryo genes, again, are shown. Blue is downregulated, red is upregulated here. So you can see which genes are in this category and which are shared with other categories like morula, trophoectoderm, or 6-cell stage embryo. And so this is very useful to see which markers, again, are most specific and enriched and make sense within the context of biology. There are also similar graphs for the gene ontology. If we look at the bottom, we see a number of upregulated terms in red. These have to do with RNA biosynthetic process and DNA binding. KEGG, local, which is actually Wiki Pathways associated genes, and we can view these genes on Wiki Pathways as we've done here. We could look at those in a moment. You also often have heat map results. When you have multiple comparisons, you can compare the z-scores across, for example, gene ontology or KEGG terms. And you have a number of other outputs here, which are very useful for further comparison. In addition to networks and heat maps, we also have visualized Wiki Pathways. These can also be found in the GoElite folder. If we look under GoElite folders and under the category, there's regulated, upregulated, downregulated genes here. If we look under regulated, for example, and now Wiki Pathways, we can see a number of PDFs here. If we open up any of these PDFs, we'll get a nice graphical display of which genes are upregulated in red or downregulated in blue. You can look at a variety of different pathways. All of these were considered differentially expressed in the comparisons that we're looking at. And you can see which comparisons up here at the top. For example, this is late blastocyst versus 8-cell stage embryo. With a cutoff, we did a 5-fold and adjusted p less than 0.05 cutoff here. So there's a lot of useful information here. This is the typical differential comparison analyses that you would normally run in AltAnalyze, but there are additional analyses you can run. Before we go on to single cell analyses, one other set of analyses I want to show you is somewhat more of the vignette analyses. And so now if you hit begin, again, you start AltAnalyze, we have human selected. We're going to select our platform as our vendor as other, and our genes, our data type as symbol again. And instead of doing process expression file, we're now going to hit additional analyses. And you'll see that this new window has a variety of different options. A lot of these options were run in the default workflow, such as pathway enrichment analysis, pathway visualization on Wiki Pathways, hierarchical clustering, principal component. Network visualization was used to make those gene ontology graphs, but we can do a lot more with this. You can translate one set of identifiers from one set to another. You can perform those lineage profile or cellular prediction analyses. You can do alt-exon visualization. We didn't do any splicing analyses in this last set of analyses because we only had gene level data, but typically we do have exon and junction level data that give us high resolution splicing information. We can generate Venn diagrams and merge files with each other based on their common identifiers. So I'm just going to show you a few things really quickly here before we go on. Actually, I'll show you principal component analysis first. First, we need to select an expression file. The original expression file we selected is fine. I'm going to hide the names of the samples, but I'm allowed to do 3D visualization here. And I'm actually going to give a name to this, and I'll say why in a second. So I run this. AltAnalyze is quickly going to perform single value decomposition analysis. And you're going to get this nice three-dimensional PCA plot, which you can actually spin around. There are numbers down here associated with the first component, the second component, and the third component. These represent the amount of variance that's described by that component. So just looking at principal component 1, you can actually describe about 80% of the data just from this first principal component in terms of how it separates the data. There's actually not a lot of separation here, surprisingly. If you look up and down to the left, from the left to the right, if you're looking from left to right and up and down, a lot of the information comes from PC3 here and even PC2. So this is a nice way to visualize your data and see how your data types points are different from each other. We didn't do any filtering to derive this plot. Now the reason I entered in PCA in that panel here is now if we go to hierarchical clustering, we can analyze our original expression file. We can analyze the whole thing. And you're thinking, well, that's a lot of genes. It might be 15,000 genes. I can also look at the filtered set of differentially expressed genes. But one cool thing I can do here now is I can actually go down, and there's a number of options here which I can talk about in a moment, but I can actually select gene set ontology terms to filter on. And one of these is actually called stored gene sets. And one of these stored gene sets is that PCA set that we just made. And so basically when we did that PCA analysis, there were top genes associated with each one of those principal components. And it's storing the top, I believe, 200 genes that are positively and negatively associated with each of those components. And it's looking about, I think, the top 5. And so if we now actually hit continue, what it's going to do is it's going to do an analysis on that expression file, even though those are raw expression values, raw RPKM values in that file. It's reading in all the values, it's converting them to log values, it's going to do a row-level median normalization because that's the default. And we can actually view these genes that are driving the differences between these clusters. This is really cool. This is without a differential expression analysis, so this is unbiased. So this is actually one form of single-cell gene expression analysis, where we can just use PCA to find the drivers. So that's cool. We can do that with other terms too. I'll quickly do it with another term. And so if we go back here, I'm going to not use stored gene sets, and I'll use, actually I'll go back to those biomarkers, whoops, those biomarkers we found earlier, and we'll look at the 8 to 10 cell stage, and we'll also look at the 6 cell stage embryo. And so we can select multiple terms here. And I'll also select pluripotent stem cells. So now it's going to select a large variety of genes, and we can visualize those too, again, relatively quickly. There are a large number of options within this heat map viewer. We're only scratching the surface, and in fact, the second set of our tutorials are going to focus just on the heat map viewer, since there are a lot of advanced features that really give you some creative ways to look into your data. So we'll just wait here a moment for this analysis to complete, and there we go. These are the genes associated with those different biological terms. And you see over here on the left, for example, you're really getting some definition here. If you look at this light blue color here, that corresponds to, it's hard to see on the screen, but morula stage. These are the different groups. The clusters are at the top. And you can actually click here, and what will happen is it will open up the program tree view. And so you can explore these signatures in a lot more detail. You can see specific genes here. And this plot should look pretty much like the plot we were just looking at. There's link outs here. There are link outs on this plot too. They're just not shown right now. But when we do some additional analyses, we can show those. We can also show enriched terms on the left here for each of the clusters. And there really is a lot we can do with this heat map viewer. But I'll come back to that later. We can also do a Venn diagram analysis. And so here I will select 2 of our GoElite results. And so if we go to the GoElite folders, and we go to, we'll just go to upregulated, and they actually automatically get moved to these archive file folders when they're done, so they don't conflict with new files we generate. So I can look at late blastocyst comparison versus this zygote comparison. And then I'm going to select an output folder, and just select our main directory there, and hit continue. And we'll actually get 2 plots. This one's a little hard to see, but this is a standard Venn diagram with the number of genes associated. Again, this is a little small. This will look a little better. You'll actually see there are only 6 genes associated with these 2 groups, 805 here with we can make this larger, the blastocyst comparison, and 204 with our zygote comparison. And these PDFs are actually automatically saved. And you can open these in programs such as Adobe Illustrator and increase the font size or change the colors if you like. There's also this nice network visualization option here too. You can actually enter in genes here at the top you want to generate a network from. You can start from a gene ontology term or another biological category that we've been looking at. And you can specify the types of interactions, where they come from, and how many degrees you want to specify between those interactions. So I'll just really quickly, instead of typing some genes in here or selecting a previous gene interaction file, we'll select a gene set. Again, I'll do biomarkers, and we can select a couple of these. So we'll select a couple of these time points. We'll select our interactions from Wiki Pathways, KEGG, transcription factor targets, biogrid protein-protein interactions, and we'll look initially at direct interactions, and we'll save these results to a folder here. Now, in addition, we can color these results based on gene expression data. So again, I'll select this late blastocyst versus 8-cell embryo comparison and see what happens. I haven't done this before. I don't know what's going to happen. I don't know how many genes it's going to produce. It may produce a lot. It may produce a little in terms of ones that are interacting, and it's a decent amount. And these are all directly interacting with each other. Here, the red arrows are indicating predicted transcriptional regulatory interactions from a database called PISAR or Amadeus. The blue lines are suggesting inhibitory interactions. In the middle, you see genes that were interacting with these but were not upregulated. A lot of these genes, coincidentally, were upregulated. Red is indicating upregulation. So this is nice, and it's suggesting that a lot of these genes are either impinging upon a central gene or regulated potentially by a common gene or interacting with a common gene, like TRAF2. You can do performing a lot of additional analyses here. Look for indirect interactions, all possible shortest path interactions. All possible interactions is useful, for example, if you just have a single gene, for example. And so let's do TRAF2, which is one of those genes we saw there. We're looking at the same interaction database as we were before. We actually have a large number of interactions here. Some of those genes are in that data set. But here's the potential interactome of TRAF2. And all these, again, are saved as tab-delimited text files and CIF files that actually can be loaded directly in programs like Cytoscape. So with that, before we move on to the single-cell analyses, I'm going to stop here, and we'll have the single-cell analysis as a second component of this tutorial. Thank you.